welcome back to the lecture series on cell culture technology. So, if you remember in the last class we talked about the defined system, and we decided upon that this class we will talk about the genesis of the defined system and why and how we could achieve it. So, talking about a defined system, we have to look into the major stakeholder of cell culture technologies. One of the biggest stakeholder in the cell culture technology in the world is the pharmaceutical industries. Why is it so? Because any drug discovery goes through different channels, different procedures. So, first of all, chemists synthesize say x number of molecule for a particular wide disease, or they isolate x number of molecules from nature or some other sources for some specific kind of pathological situation. So, then they needed to do the testing. Of course, they cannot directly go to the animal testing. They have to go through a cell culture or testing in the cells in vitro. So, the first testing, so if you just let me start putting them down, so that okay, today we are into week 5, lecture 2, W5 L2. Okay. Now, where we are starting today is genesis of the defined system is one. Second, how, of course, why there, which is essentially answering why there is a need for defined system and how we could achieve the same. Okay. Now, as I told you, we just switch gears and told you the drug discovery industry, drug discovery process. Heavy dependence on cell culture. Cell culture trials before moving to animal or in vivo trial. And these are all in vitro trials. So, now since I have jotted down the thing, how you should think over this. So, this needs bit of a thinking process, what we really meant by that. So, say for example, we wanted to prove a point, let us talk about that. We say that a system is working towards a medicine which will say prevent Alzheimer's. Fine. Now, you have to test this. If you have to test this, you need it first of all in vitro setup. So, let us jot down the points. So, this is how I am going to proceed. Say for example, industry. So, we will have a case study which will be an imaginary case study, but that imaginary case study or hypothetical case study will help you to appreciate what we meant by the defined system. So, we will be coming to the defined system from a different angle. So, bear with me. So, say for example, drug discovery process. Here is the drug discovery process, and in the drug discovery process the goal is now to screen some X number of drugs against Alzheimer's disease. So, let us enumerate what all you will be needing. So, of course, we know the process is there has to be an in vitro trial, there has to be an in vivo trial, and it should follow that sequence. You cannot, and remember, you cannot jump to an in vivo trial without doing the correct in vitro trial. Now, what was existing in the industry is something like this. When you talk about screening Alzheimer's drugs, you will see many work. What they will do, I will tell you. They will, okay, before I get into this, those who doesn't know what happens in Alzheimer's, so you lose the hippocampal neurons of the brain. So, if you look at the brain, so here is your brain, this is the dorsal view, you are looking at the brain from the top, and here is the ventral view of the brain, where you are looking at the brain from the ventral side. This is the ventral, and this is dorsal, and out here is this position slightly deep inside lies hippocampus. You really need good surgical skills to isolate the hippocampus. You cannot do it so easily. You really need to go down and pull out this tissue. Now, what happens in Alzheimer's, for those who are not aware of it and for those who knows it, these cells which are part of this, which are essentially interesting neurons with a very nice pyramidal geometry. Their cell body looks like pyramids, something, and they are large cells, and so this is how most of them looks like. If you look at the cell body, it will look more like a pyramid like this. Okay. So, there is a three dimensionality into the structure. Okay. So, these are called pyramidal neurons. So, these pyramidal neurons form a series of circuits within this structure which are called CA1, 2, 3, 4, and of course, there is a zone called dentate gyrus, and these pyramidal neurons starts to die at a certain point in life in Alzheimer's. Mostly these are age related disorders. So, when we talk about Alzheimer's, we talk about age related, mostly, again this is mostly because age related neurodegenerative disease, and this age related neurodegenerative disease means these aging neurons, in other words, as you are aging, the neurons are also aging. These aging neurons dies for an unknown reason. Okay. So, now you wanted to screen a drug for these aging neurons. What paradigms followed in many places, and some now they are shifting. So, I will tell you the existing paradigm for. So, this is your case study. So, now let us see what are the paradigms which people are following. So, the first thing what you will be needing for this is, one, you need hippocampal culture. So, which has to be a primary culture, because there is nothing called hippocampal cell line or something, because these are completely differentiated cells, hippocampal cultures. Then what you need is a defined medium. I will come to that why. Then you need a defined substrate. Fourth, define growth conditions. These are the most optimal things what will make this a story to explore further. So, what people are doing now, let us see what is the current status. So, the current status is, now I am putting here on this side. Okay. Now, you have the hippocampus culture, which is people use embryonic day 18th rat or mouse. What does that mean? So, a rat or a pregnant rat or a pregnant mice goes through a pregnancy phase of 21 to 23 days, somewhere in between. On 18th day, the fetus. Okay. So, there are two words which are being used. So, when they go through this whole cycle of 1 to 21 to 24 days. So, up to 14th day, this is called an embryo, and from 14th day till birth it is called fetus. Okay. So, this is the cycle a rat or a mouse goes through. Now, on day 18th, somewhere out here, 17th, 18th, the hippocampus is fully developed in the fetus. So, what you do, you take the mother mice or rat, you sacrifice it. So, let me go to the next slide, that will make more sense. Okay. So, mother mice or slash rat sacrifice the subject. Now, how you are sacrificing is important. Are you using CO2 or using chloroform or using cervical dislocation has to be fixed. That methodology has to be fixed, which methodology you are using. Then post this you isolate the fetus, then you dissect out the brain, then you pull out the hippocampus, so here you are having the brain, and as I told you that is the dorsal side and here is the ventral side of it. And here is your concerned tissue. So, you really needed a very good pair of hands to pull out the hippocampus. Once you pull out the hippocampus, it is more or less it looks like this. This is we are talking about the fetal hippocampus, something like this. Though this word hippocampus had come from, the meaning is the seahorse in Greek. Okay. So, its shape is like a seahorse. So, that is why it got the name. So, here is your isolated tissue, hippocampus. Now, this hippocampus, what you do, you dissociate this tissue. What I meant by dissociation is that if you imagine this tissue, it is something like this. It is a network of neurons, pyramidal neurons, very small, tiny pyramidal neurons. It form a huge network out there. Now, when you are dissociating it, what you are essentially trying to do, so in between you have a lot of extracellular matrix features. I am just giving 2-3 neurons here showing you, but there are thousands and thousands of them forming this tissue. So, what you do using some XYZ technique out here, so you kind of split the cells into a single suspension. So, what you wanted to achieve is you have the cells separated out like this. But while doing that process, most likelihood you are going to break some of these. So, what you will be getting as a single cell suspension, most of them will break, you will be getting like this. These are the individual cells which you are going to get out, and this is called single cell suspension. Now, when we talk about this part, how you are going to do this, you have to realize that your options are two. Either you mechanically dissociate them, mechanically dissociating the tissue. So, you can mechanically dissociate the tissue because these are very soft. What you can do is that you take a pipette tip and you just cut the tip a little bit. Just say for example, you have a pipette tip like this. So, you cut it. So, what you are left with is something like slightly wide bore. So, you have your tissue out here in the medium. So, along with 1 second, okay. You take the tissue in, out, in, out, in, out. In that process, what will happen? You will be obtaining what we call as single cell suspension. So, it is a kind of a shear force by virtue of which you are breaking the tissue. It is a very brute force method, but this brute force method does work for tissue which are soft, okay? And that is good enough, especially for hippocampal neuron, that is good enough to do this. So, what I will do is, here I will close in, and the next class will continue from here to explain that how this tissue will be plated, because see, in this process what we are happening, if you go back the slide before, we talked about cell plating rules, and here I mention about age isolation rules. So, we are talking about the isolation rules now. Okay. So, this is your map which you have to keep in mind. As long as you could follow this map, you will be able to follow these lectures very smoothly. Okay. So, I will close in here, and we will continue with this hippocampal thing, and we will talk about what are the challenges what the industries are facing. Okay. So, we will just stop here. Okay. Thank you.